Be right back. First, any questions or concerns? So let me introduce you to these case studies. And one of these is relatively easy. The other one is a little harder. We're not going to be able to answer all the questions on the second one, but we will be able to answer some of them. When I give you a case study and ask you to answer the questions on your own, I will expect that you can find the materials within PowerPoints and or your book, and you can find the answers there. I will select certain questions that I know that between your PowerPoints and your books, you should be able to find the answers. And like my students from last semester who had me for lab, sometimes they'll span multiple chapters. For example, I gave them one about the nervous system last semester that also asked them to define what an ion was from chapter 2 and what a membrane potential was. This one, I don't think it's going to span like that. We'll have to look at it, and you can think if there's other materials, things that you know of that would help answer these questions. And some of the questions I'm not going to expect you to be able to answer. I'm not even sure what they mean by some of them. But the majority of them, yeah, we're capable of answering them. So, and what I want you to do is we'll discuss all of the questions, and I'll tell you which ones not to answer and which ones you're not expected to answer. But then I want you to send a write-up answering those questions that I'm asking you to answer in your own words. So I'll let you know which ones I'm asking you to answer. So not all of them, but there are a lot, the majority of them we should be able to answer. Any questions so far? Go ahead, Becky, go ahead.
Sorry. So we're just going to go over this to see what's going to be up for the future, like in the future,
yeah,
right?
You'll see kind of what I'll expect in the future. Some of them, overall, this is where they're going to come from, but you'll understand what we're referring to when we talk about a case study. So we're looking at a real case scenario and applying the knowledge that we have to answer the questions and possibly figure out what's going on, depending on what the situation is. And so when you submit them in the Dropbox folder right here. And I need them submitted as a JPEG, a PDF, or a Word document. If you submit them as some other type of file, I may not be able to open them. I can't open Google Docs. I wish I could. Google Docs. I wish I could, but I don't have the software to open them, and I would have to pay for the software to open them. And right now, I'm not willing to do that.
Okay, so Dropbox for the assignment is JPEG,
Right.
JPEG, PDF, or
Yep.
is that it?
Yep.
Okay,
And
no,
it's in the syllabus how you should submit that kind of thing.
got it.
All right. So let's jump into this. And for all I know, and from all you know, you might find this the most interesting part of the class, because I know sometimes they are. Bear with me as I get rid of some extra tabs. Because one thing that we're going to have to do for some of these answers that are easily answerable if we look them up is go to Google and see how Google describes something. Just getting rid of all my tabs up here. So the first study is about blood. Not that one. So in this study, they're going to give us some numbers. The numbers for what's normal values are in this second link. So let's look at the case study and see what it says. So this is a case study about iron deficiency anemia. And I'll expand it out and read it to you. Give me 1 second. So Dolores Wellborn is a 28-year-old attorney living in Portland, Oregon. She's in the second trimester of pregnancy with her first child. And though her pregnancy had been progressing normally, recently she has noticed that she tires very easily and is short of breath from even the slightest exertion. She also has experienced periods of lightheadedness, though not to the point of fainting. Other changes she has noticed are cramping in her legs, a desire to crunch on ice, and the fact that her tongue is sore. She doubts that all these symptoms are related to one another, but she is concerned, and she makes an appointment to see her physician. Upon examining Dolores, her physician finds that she has tachycardia. Tachycardia means you have a higher heart rate than normal. If I remember right, I think it's consistently above 120. Pale gums and nail beds, and her tongue is swollen. Given her history and the findings of her physical exam, the physician suggests that Dolores is anemic and orders a sample of her blood for examination. The results are shown in table 1. I want to explain something, and we'll understand this more in a couple of days, but I'm gonna stop this just for a minute and explain why does she have a higher heart rate. She has a higher heart rate, and the reason that her anemia first off, what's anemia? Before I explain this, what's
Low
anemia?
red blood cell count.
A low red blood cell count. That's right. What else could that be saying about her blood? If her red blood cell count is low, what else is maybe there?
Oxygen.
low
Ox-
hemoglobin.
oxygen and her is possible. Her, and that could be explained why she's tired, right? If she's struggling with getting oxygen in and out of her cells and she's short of breath, she's getting tired. And it's because she's probably a little hypoxic. Another thing that if her red blood cell count is low, her overall blood volume is probably low too. And what that means, okay, let me start. Now that I think about it, the other reason she could be tachycardic is because her oxygen levels are low. Because if you can keep the blood moving into the lungs, if you remember from last week, it goes into the lungs, it comes back into the left side of the heart before it goes out to the rest of the body. So it gets oxygenated and then it goes out to the rest of the body. It may be her heart's way of helping with oxygen perfusion because she's already low on oxygen. However, the main reason that someone would be tachycardic is due to a low blood volume. And that is related to this, as my cat walks across the front here. Cardiac output equals stroke volume times heart rate. This is a great equation. What it says, cardiac output is defined as so cardiac output is defined as the amount of blood pumped via the left ventricle, but remember from last week that the right and left ventricles pump the same amount of blood per minute. So the amount of blood pumped by the heart, by the left ventricle, per minute. The stroke volume, this SV, is, bear with me here, is the amount of blood pumped or ejected from another way, amount of blood, bear with me, I'm going to change this cardiac output definition. The amount of blood pumped or ejected the left ventricle per minute. So how much blood is actually going out to the rest of the body per minute from the heart? The stroke volume is the amount of blood ejected or pumped from the left ventricle with each heartbeat, each heart contraction. Heart beat. And the heart rate, as you probably already know, the heart rate is the number of beats or contractions per minute. Beats or contractions, heart contractions. Per minute. So what this means is that if you typically have a stroke volume of 70 milliliters of blood, seventy milliliters of blood is your stroke volume. And then you have, let's say, your typical resting heart rate is, let's say, 72 beats per minute. Times 72. You times those together, beats per minute, BPM. What that means, I'll grab my phone. Yeah. Actually, I'm going to do this differently. Beats per minute. So 70 times 72 means that the cardiac output of the heart per minute is 5,040 milliliters of blood per minute. Does that make sense? So the stroke volume is the amount of blood ejected from the ventricle with each contraction. And the heart rate is the number of beats per minute. And typically we have about 5 liters to 5.5 liters of blood in our bodies, right? Now, our cardiac output doesn't tend to change a lot. It can change if we get a lot of exercise, which will allow us to lower our resting heart rate if we can increase the volume of our heart and increase the amount of contractions and the force of the contractions through exercise. However, it doesn't change quickly. It's years to months of exercise. I don't know how much exercise, just it requires exercise to be changing your cardiac output. Your stroke volume can change fairly quickly. Your heart rate can change fairly quickly, right? Your heart rate can go from being 72 to 100 if you decide at that moment to drink an energy drink like I drink. It doesn't take much. So if her stroke volume, if she's anemic and her stroke volume is dropping for that reason, because she's got lower blood volume already, And she has to maintain the same amount of cardiac output. It's your body's way of survival. It doesn't change. Cardiac output doesn't tend to change. It can change temporarily, but it doesn't change for consistently over a long period. So let's say, for example, she's a little dehydrated, so now her stroke volume is 50 milliliters. Well, she still has to maintain a cardiac output of 5,040 milliliters. The only way to compensate for that is to increase her heart rate. Do you see it? If this is a constant over here, if her stroke volume changes because her blood volume is going down because she's anemic, or you get another way that this happens is you get dehydrated. The only way to compensate for that and maintain a cardiac output that will keep you from dying is to increase the heart rate. Because this is a constant. So you want to think of this as a constant. For right now, think of this as a constant. If this goes down, how are we going to keep this a constant? You get dehydrated, so the amount of blood in your body goes down, which is going to change the stroke volume. But you still have to maintain the same cardiac output. So the number of beats is going to go up. Because if this goes to 50, say this right now, this is 5,040, the cardiac output, 5,040 milliliters. And we're getting sick. And we're dehydrated, we're anemic, and our stroke volume is down 50 milliliters because we've just had a lower in blood volume. The only thing that we can do, that our body can do at that moment, is increase the heart rate to keep this the same. So and the way, so this would be HR. How do we solve for HR? Take this number, 50, and divide it by this number. And that tells us what her heart rate would be with a stroke volume of 50 milliliters. Make sense, Zena?
Kind of, because how do you measure the stroke volume? Like, is
You
that
really can't. You kind of have to estimate it. There's no way to really measure that.
Oh,
But
okay.
if she's anemic, chances are she's got a lower blood volume. Does that make sense?
Yeah.
If she's dehydrated, you're definitely going to have a lower blood volume because where's that water? How's that water leaving your body? You've had a lot of water leave your body. What is it going to do? It's going to lower the blood volume.
Right.
Because Zena's the only one that I can see right now, and she's looking a little confused. Now I can see Becky. And that's why I'm when I look at her and she looks like she might be confused, then I know that probably other people are confused.
Yeah.
So that's why
we're going, I'm sorry, I
Go
didn't mean
ahead.
to cut you off, but
No.
I'm assuming we're just going off of like the basic normal, what we consider is normal for
Right.
men and women, which was the 5 liter and 5.5.
Normal is about 70 milliliters of blood as a stroke volume. If people who are athletic, how they have a lower resting heart rate I may have mentioned it. So I really liked Lance Armstrong until I realized what a booger he was, because I liked to cross country cycle and I really admired his skills. He had a lower resting heart rate of around 50 at one point. And what that means is that his stroke volume has gone up, allowing his heart not to have to work as hard per beat to maintain the same cardiac output. Because if the stroke volume goes up, the heart rate can go down. And so it works both ways. Michael Jordan at one point was noted to have a resting heart rate of 40. My mom has always had a lower resting heart rate. My heart rate is always high. Part of it is because I'm chronically dehydrated. And part of it is because I drink too much caffeine. All right, so coming back to the case study, coming off this because we'll see a lot more of this in a week. Coming back. So diagnosis of anemia is due to iron deficiency and oral iron supplements are prescribed. Dolores' symptoms are eliminated within a couple of weeks and the remainder of her pregnancy progresses without difficulty. These are her blood sample results. Her red blood cell count is 3.5 million per millimeter squared, which is about, oh, well, before I say something stupid and I say something wrong, I'm not going to answer that. Her hemoglobin is about this, 7 grams per deciliter. Her hematocrit is 30%. Where should her hematocrit be if she's a female?
45 to 50, I think it was.
I think it's a little lower than that. It's about 42 to 37. But yeah, this is, I mean, if you think that's almost a fourth lower, right? That's kind of a big deal. Approximately a fourth less than she should have. Her serum iron content is low. Her mean corpuscular volume is low. What this means is the size of her average red blood cell is small. It's lower than it should be. The volume of her individual red blood cells are smaller than they should be. The amount of hemoglobin per vessel for her cell, red blood cell, is low. But the total iron binding capacity in the blood is still high. So now, somebody do me a favor and write down these numbers. Or I will write them down too. RBC is 3.5 million. Per millimeter square cubed. Our hemoglobin is 7 grams per deciliter. Her hematocrit is 30%. And her mean corpuscular volume is low. Low. Her mean corpuscular hemoglobin concentration is low. MCH, MCHC is low. Her iron capacity is high. So now, if we have these numbers and we don't know what they mean, there's a really great chart. So we click out of this. And right here is our complete blood count reference, because that's how they assess those results, is they took a certain blood volume from her, maybe a couple milliliters, and then they did a complete blood count. Looking at all differences in the red blood cells, her white blood cell counts, everything that they could possibly do, they did. So her red blood cell count for females, if you look at this sheet, I'm gonna enlarge it a little bit. Okay, for males, typically the red blood cell count is 4.3 liters, 4.32 to 5.72, 10 to the 12 liters per liter. For her, she was at, what was it, 3.5 million.
Mhm.
And this, what is this value? 3.5 million. What is this value? This is pretty high, but hers was millimeter cubed. How many millimeters are in a cube? 1,000. So there's 1,000 milliliters in a liter. Actually, you're even going to go lower than that. Bear with me here. Let me think here. If they did millimeters cubed, this is 1 liter. So the way you'd figure this out, and I didn't even think about this until now, but her value was 3.5 million per millimeter cubed. They're putting it in liters. So the way we convert this down is 1 milliliter or let's actually put it this way, 1,000 okay, I gotta type, I can't. 1,000 milliliters equals 1 liter. Okay. And because, and one milliliter is equal to 1 centimeter cubed. How many millimeters are there in a centimeter? If you look at your typical yardstick or your ruler, how many millimeters are there in 1 centimeter? Somebody tell me. I know you can calculate this out, or you can look at a ruler in your house.
Like, isn't everything like divided by 10?
It is. So one centimeter equals 1 millimeter. Or shoot, that I know that isn't right. 10. It sounds great until I say it comes out of my mouth and I know it's wrong. 1 centimeter equals 10 millimeters. So what that means is that every centimeter cubed, 1 centimeter cubed equals 10 . Is this right? You know what? And I'm gonna, and actually I was almost was wrong. It's 10 times 10. Times 10. You know what? How do you measure volume of something?
It has a formula.
So what's the formula, Claire?
Hold on. I should know this.
You guys
Formula.
you didn't expect to be doing math during this case study, did you?
Hold on, I'm getting it. V equals pi r squared h.
Okay,
So
let's not do that because that's for a circle.
over
Let's not do that first. Let me just tell you what it is. Length times width. Length, that was funny, times width times height.
I was giving you the volume of a cone too.
Oh, okay, well, thank you. All right.
Sorry, I mean,
Times height. So 1 centimeter cubed equals 10 millimeters. So to figure out how many millimeters are in 1 centimeter cubed, equals 10 times 10 times 10. So 1 centimeter cubed equals 1,000 millimeters cubed. Does that make sense? You got this? Maybe. So now, if we know that 1 centimeter cubed equals 1 milliliter, and you guys, I'm doing this because of the numbers they gave us and how to compare up the numbers, okay? I'm not expecting you to do this on an exam. I'm just showing you how to do it. And then when you go into your next classes, you'll learn a lot more there. So 1 centimeter cubed equals 1,000 millimeter cubed. So for every 1 centimeter cubed and 1 milliliter, okay, for 1 centimeter cubed equals 1 millimeter. Or no, just a sec. Cubed. All right, bear with me. We're just
I'm
gonna.
just happy we're going over this part in the afternoon, not first thing this morning. I would be totally baffled.
I thought you guys were more morning people.
Oh, yeah.
I'm not really either at this point. I'm kind of an exhausted pigeon. Let's see here. 1 centimeter cubed equals 1 milliliter. And 1 milliliter so this actually, let's say this. So 1 milliliter, 1 centimeter cubed, 1,000 milliliters equals 1 liter. So if we take this number right here, 1,000 millimeters cubed, and times it by 1 mil, so times it by 1,000, we're at 1 liter, right? And so within every, now I'm just getting myself confused. Why did I even do this? Let me think here. Because this isn't making sense according to what her numbers are and how it would work out. 3.5 million per mil. Oh, okay. So 1,000 times 1,000 is what?
$100,000.
Is it 100,000 or is it a million?
It's a
1,000
million.
times 1,000 is a million.
A million.
That's a million.
Okay. So what we have to do to bring, why am, I'm not happy that I keep losing my numbers. Okay, one more zero. One. Okay, there's a million. So what we have to do is take the numbers that we're given here over there and divide them by 1 million. And then we have what the value is. So
So
this,
it's 3.5 then. Right?
probably,
Yeah, because a million one divided by 3.5 is going to be 3.5.
everybody agree with her? I'm just gonna say,
Well, one divided by 2 is 2.
Yep.
So 1 million divided by 3.5 million is going to be 3.5.
Yeah, it's definitely 3.5.
All right, we're going with what you said. But that's the way you break that down. And I know that was a lot more work than I thought because I didn't plan on, this chart looked really great until I just tried to apply it to that case study. I did some other case studies for my physiology class in Stevenson. And we used this chart, but we had numbers that matched with the units. And this, obviously that it didn't work here. Okay, so is it low?
Yes.
Yes, it's low. Going on the hemoglobin,
Can you go back to the other screen with the,
well, let me tell you what the number is because I did write it down. 7 grams per deciliter. For females, it should be 12 grams to 15.5. Is it low? If it's 7? Okay,
yeah,
so
the average 12, yeah, so it's low.
it's low. Her typical hematocrit, like, and you know what, I think someone, well, that's males, Mary. All right, for females, should be 34, according to this, it should be 34.9 to 44.5. So is she low?
Mm-hmm.
Okay. So that's the way they diagnose the anemia first. What they probably, the easiest way that I would diagnose it first is I would look at her red blood cell count and her hematocrit. And you know right off if it's low, right?
Mhm.
Mm-hmm.
So come back after that real mess of numbers. Describe the structure of hemoglobin and explain the role played by iron in the transport of oxygen. What was the structure of hemoglobin?
What do you mean by what's
What
the
did it
structure?
look like? What did it look like?
Like the red blood cell, right?
Right, it's in the red blood cell, but what does it look like? If I put a little bit more effort into these case studies, I would have had a PowerPoint, and I don't have one here. You know what? I think I might, no, let's do this. Hemoglobin. What does hemoglobin do? What's in it?
Isn't it the fragment? Oh, that's platelet. Never
Yep.
mind.
So it's got 4 polypeptide chains. If you go back and you look at the blood chapter, you'll find this information. But I'm not going to search through the PowerPoint at this very moment because it means that I have to change and flip screens around. So if you look at that PowerPoint, we have 4 polypeptide chains, and within the center of each one is a heme group that contains an iron molecule. And that iron molecule is capable of binding oxygen. You see that?
All I can see is your whiteboard.
Oh, shoot, darn it. Give me 1 second. Can you see it now?
Yep.
Zena, why didn't you say anything? She's like, you'll catch on eventually that we don't see anything. All right. So 4 polypeptide chains. This is hemoglobin with a heme group in the center of each one. And within each of your hemoglobin molecules, I'm trying to remember, there's at least 250 million hemoglobin molecules within each red blood cell. So without the iron in the heme group, you're not binding oxygen, right? So if you're iron anemic, that's the reason your hemoglobin levels are low, because first, you're not going to make hemoglobin if you don't have that iron either. Even if you made it, it probably wouldn't stay together as a structure because the heme is needed for the structure. Does that make sense?
Yes.
Maybe. So let's see, what else did it say? Okay. So the first one is describe the small molecule of hemoglobin and explain the role played by iron in transport of oxygen. That's your answer. It's 4 polypeptide chains, each with a heme group in the center that contains 1 iron. Each iron molecule is capable of binding an oxygen molecule. Now, how is iron stored and transported in the body? And what is iron deficiency anemia, and how does it occur? And what are the most common causes? So first, how is it stored in the body? And I am
Stored in like the liver or the spleen.
It's stored in the liver, and how is it transported? I'm going to go back to our chapter 18 PowerPoint now that I think about it. so this would be where you can also find the structure of hemoglobin. So you see why I said that when I give you a case study, go back and look at the PowerPoints. Because if I haven't mentioned them in the PowerPoints, I'm not going to ask you to give me an answer about it. I'm going to be, these case studies are a little harder because I haven't taken away questions. All the questions came with the original case study. So changing here, here's how it's transferred. So it's like, so this is iron metabolism. Is bound to transferrin, and it transports it throughout the body. And like Becky said, it's stored in the liver. And it's stored as a ferritin molecule. I wouldn't ask you to give me those numbers because you can see that I had to figure out how to do those numbers. And we've never just, if we hadn't had discussed them, or I haven't put it into a PowerPoint, I'm not expecting you to know it. Any questions about that question? Let me go. Let's go to the next one. What was the next one? Do you think these are kind of interesting? They're like, no, because we're brain dead.
Both.
Okay. All right. Going to, then now.
I honestly like these case studies more because I feel like with the labs, if we're just doing like the blood experiment and stuff, like that's something we're never gonna do in real life. Well,
Right.
not all of us, but with these
Right.
case studies, we can just apply it to like real life.
Right.
I
Exactly.
agree.
Exactly. That's what I think they're fun too. What is iron deficiency anemia and how frequently does it occur? Well, I would, if I were you for this answer, I would look in the textbook.
So I actually have
Go
all these
ahead.
questions. I have all these questions done, but I don't see anything about the frequency.
Is it ask you about the frequency of it?
Yeah, I didn't see it in the
Oh, how
textbook.
frequently does it occur? You know what? What I would do for that one is I would Google it and cite where you got the information. Because that's honestly going to change from year to year as new statistics come out, right? And honestly, that's not something that, that's what I would do, iron deficiency. Statistics on IDA. IDA.
If we cite it, do you want APA or MLA?
Oh, I don't care. I don't care. Just make sure it's cited. I don't care how you write it up, as long as it's in a Word, PDF, or JPEG, and I can read what you wrote. No, don't worry about that kind of thing. I don't want you to write me an essay, and that would be APA. I don't want that. Don't make it that hard. IDA. And I'll bet you money this is not IDA. Anemia, well, that is. Okay. Let's see what this says. So this is the worldwide organization. It is estimated that 1/3 of all women of reproductive age are anemic. Iron anemia. Is there a way of searching? Yeah. IDA. I'm gonna do this rather than what I wrote in there. IDA stats. IDA statistics. And I honestly, if I was looking for some, and I maybe not this, percentages. If you can find it on the World Health Organization, that's by far your best source. And of course, we can't find it anyway. Occurrence. Nope. Okay, giving up with that one. If you could find the answer there, that would be a good source, but obviously I can't find it there. Let's go and look somewhere else. Okay, get rid of this altogether. IDA occurrences.
There's a good article on aafp.org.
Yeah, what does it tell you?
It says the prevalence of iron deficiency anemia is 2% in adult men, 9 to 12% in non-Hispanic white women, and nearly 20% in Black and Mexican American women. 9% of patients older than 65 years old with iron deficiency anemia have gastrointestinal cancer when evaluated.
Okay. So I would, and you know what, and because I'm hearing impaired, I only got so much of that. If I read it, I would understand it a lot better. Where did you find it?
It's the aafp.org, and the article is just called
I'm sorry, AAFP.org.
fp.org, and it's iron deficiency anemia. That's just the article title.
Okay. Oh, that's cool. See, I wouldn't have known. I wouldn't have know what to search there. Iron deficiency. And of course, I spelled it wrong. Where was it? Was it this one right here? Yeah, that's what.
Yes.
And if I was going to answer that question, and yes, just cite it where you got it. I'm honestly not going to look to find where you got it and go to that source to grade it, but cite where it was. Questions about that one? Thank you, Lauren. That was easier. Now, what are the most common causes of it? What do you think?
Vegetarian, pregnancy, menstruation.
So most of it is diet, right?
Oh my gosh, never mind. I'm so sorry.
No, you're all right. You're all right, but you're right. You're right. Let me make it. I mean, I'm really good at giving really long answers. And so you're right. But let's say diet and menstruation. So first off, most common causes diet. Why are women more prone to IDA than men? Well, menstruation. What are the red blood cell indices and what tests are diagnostic for IDA? The red blood cell indices are tests to assess red blood cells in a complete blood count. And this is another answer I would just Google and put a citation. And I would say that these are the tests that would assess for iron deficiency anemia. First off, the number one is the hematocrit. We understand the hematocrit. And then there's other tests. How is it treated and how is it prevented?
Routine blood work.
Routine blood work is how it's treated.
Prevented.
Prevented. I guess so that you would know.
Yep. I mean, that's how I would assess it, but how I would treat it and avoid it is taking an iron supplement.
Yep.
But that's the end of this one. Any questions about it?
No.
So let's go to the next one because it's a little bit more complex too.
Do we have to do all the math?
Not on that one, no. I'm not expecting you to do any of it. And none of those questions actually asked you about math. Did you notice that?
Yes, it was just comparing it to the chart.
I just thought we should make sense of those numbers. Yeah. I mean, if I'm given numbers, I want to be able to make sense of them. And as a group, we could do that. I wouldn't make you do it individually. So CAR T immunotherapy, engineering the immune system to fight cancer. Jesse's cancer was back. He'd been diagnosed with B-cell acute lymphoblastic leukemia, or BALL, and had gone through multiple grueling rounds of chemotherapy. At the time of his initial diagnosis, he was ready for a fight. He was determined to overcome the illness. His oncologist, Dr. Claire Sanguine, was hopeful for a full recovery after detecting a decrease in the number of cancer cells. Sadly, however, the most recent test indicated the cancer had returned. Now, all Jesse felt was despair. Nevertheless, Dr. Sanguine was still hopeful. She felt that Jesse was a good candidate for a new treatment called CAR T therapy, which had recently been approved to treat BALL. Now, what in the world is CAR T therapy? So how is CAR T therapy different from traditional chemotherapy? What you guys, let's hypothesize, let's suggest, what are the possibilities here? What do you think, from what we were talking about today in class, what do you think this might be? What?
The second
Well,
round of chemo would go better because of the memory cells or whatever.
That's a possibility. What would the memory cells be against? Help me out here. What do you think are the memory cells in that case? Now, I honestly, up until this moment, didn't know anything about CAR T therapy. And so this isn't a question that I would give you on case study.
So is it the T cells that would be the memory cells?
Yes, yeah. Will they develop memory? Are you sure that they're going to develop memory cells? Because these are bioengineered.
No.
Yeah, and I'm not sure either. But look right here. One of the first questions asks, guys, first off, what would you think this therapy is? What's traditional chemotherapy? I know we have some smart people in here. You guys are all smart.
Isn't it
What?
where like they basically take like radiation and the radiation like kills off the cells, like the cancer cells, but like it doesn't really differentiate like the difference between like normal cells and the cancer cells?
Right. Specifically, it's radiation is something different, but your chemotherapy is, like you said, Martin, you're giving someone chemicals that's intentionally killing activated cells that are mitotically active. And it's
Doesn't it,
not, go ahead.
sorry, doesn't it also kill like the white blood cells or was it leukocytes or whatever you want to call them?
Yeah, it's going to kill all of your B cells.
Yeah.
It's not going to go after specific ones. It's going to kill all of them. So how do you think this treatment might be different? What do you know about T cells? What do you remember about T cells from this morning?
They involves the production of lymphocytes.
And how do they do that? How do they get activated? Let me ask another question. What part of the immune system are they part of? Generally speaking, what part do we think that they're part of? Talked about two different types of immunity. What part were T cells part of?
Adaptive.
Adaptive. So what does that mean?
It means they originate from your body and adapt to the environment. I don't know.
How do they adapt?
Somebody else.
You're right. How do they, what about them makes them specific?
They create like go ahead.
No, it's fine. I don't really know.
Zena, what do you think?
I think they create like specific antibody to that antigen.
Okay, it's not
And
the
those
antibodies.
cells?
It's not the antibodies because those are B cells. We're talking about the T cells right now.
The T cells.
Do you remember that they have specific receptors on them for specific antigens? Do you remember that?
It's ringing a bell, but it wasn't in my forethought.
Okay, let me back up just a little bit. And you guys, if we don't answer all the questions, that's all right. Or if we don't even make it through all of the case study, that's all right. I just wanted to discuss it and we get as far as we get. And I'm only making you responsible for as much as we get to. So looking at this, Bear with me here. And this isn't a case study I would give you, because I think it's harder than the ones that I would give you. But that's also why we're discussing it. So a T cell only gets activated when it binds an antigen. That's the reason that this therapy is better than your general chemotherapy. These T cells are only going to kill other cells when they bind to the right substance, right? You remember that about cytotoxic T cells? They only kill things when they bind to the right receptor.
So basically, they're not just going to go in and kill all the cells. They just kill
Right.
the one.
Right, right. Now, looking at, I don't know why it made me go all the way to the beginning here, but it did. So how is it different? It's because they're T cell therapy. They're only binding to specific antigens, and that's what activates them. If you look here, and I'm going to zoom in on this. This is how they describe how this works. So here's the picture. Okay. An engineered T cell recognizes and attacks a leukemia cell. The CAR molecule binds to CD19. Do you remember what I told you CD was earlier today, what it meant?
Cell differentiation.
Right. So this is specifically a marker on leukemia cells called CD19. It's specifically an antigen on leukemia cells called CD19. This activates the T cell, which releases perforin. You remember that from earlier today, forming pores in the cell surface. And then granzymes, remember those from earlier today, enter through the pore and initiate apoptosis to kill the cancer cell. See how this relates to what we were talking about earlier today?
Mm-hmm.
Kind of fun when you realize it's not just a bunch of dumb stuff that doesn't make any sense. So we know what the T stands for, right? For Jesse's treatment, we're going to look at this a little bit more. Bear with me. Why is the source of the T important? All right. Okay. Okay. Let me ask, where do you think those T cells are coming from? Look at what it says. For Jesse's treatment, where will the T cells come from? Where do you think they're going to come from? Where do they originate in the body?
Bone marrow?
So I would assume that they're going to culture them from the bone marrow. Why is the source of the T important for organ tissue rejection? Do they have to come from him, or could they come from anybody?
Well, it would be best to come from him.
Why? Why, Becky? Because I think I could give him some bone marrow pretty easily. Why does it
Right.
have to come from him?
They do so your body doesn't reject it. So
And what structure did we talk about this morning that would be involved in possibly causing a rejection where his body wouldn't like my T cells? I thought my T cells were kind of nice to start out with.
is it the epitope?
No, you're close. What else could it be?
Antigens?
You're close. What else could it be?
That's all I got.
No, I know you got more. What else could it be? Someone want to help her? What else
it
could it be?
You know what is it? The immunocompetent?
Nope, it's not that. You're
Okay.
close though.
Is it your antibodies?
Say it again.
Is it your antibodies?
Nope, but you're close. What was the carrier protein for antigens that we talked about this morning that was first identified in because of transplant rejections? You
It's not
remember that?
Glycoproteins or the MHC?
Yep. Yes, the MHC. Good
What?
job. Booyah. Booyah. Okay. So MHC, it's important that those MHC molecules don't cause tissue rejection for him, right? Because that's how your body recognizes self from non-self is via those MHC1 molecules.
So I have a question. Is that why if somebody they test like to see if you're a match for like bone marrow transplant, stuff like that? So they say family members are better because you have more of the same DNA.
They're looking for MHC matches.
Okay.
That's having never done that and studied into it, that's what I would think that they're looking for. I haven't done a lot of work and research into it, but that's what I would think. Because they're the most your MHC molecules are so unique to you that unless you have a twin, nobody else has the same structure. And so what they're trying to get is the best match possible. What does the CAR stand for? Now, it doesn't say here what the CAR stands for, but I looked. CAR stands for chimeric antigen receptor T cells. That have been genetically engineered to produce an artificial T cell receptor for use in immunotherapy. Let's look somewhere besides Wikipedia, because sometimes Wikipedia may not be right. let's look at cancer.org. So CAR therapy is a way to get immune cells called T cells, a type of white blood cell, to fight cancer by changing them in the lab so that they can find and destroy cancer cells. CAR T cell therapy is also sometimes talked about as a type of cell-based gene therapy because it involves altering the genes inside someone's T cells to help them attack the cancer. Let's do, I'm going to do some more. I'm going to bear with me here. I want to look and see what other questions we have, and then I'm going to come back to that again. Okay. We don't really need more of that information, but know that what it's doing is it's a genetically engineered antigen receptor. So you've genetically changed the cells to give them a different antigen receptor that will allow them to bind to CD19. And when they bind CD19, they'll kill it. Is that clear to everyone? Jesse has a type of, oh, and let me ask one more thing. Are they cytotoxic T cells or helper cells? What do you think?
I think helper cell.
Say it one more time, Toyin.
I said helper T cell.
Or apoptosis cells, so they're cytotoxic. I would say that too. Jesse has a type of cancer called B-cell acute lymphocytic leukemia. What is the normal function of the cells that give rise to this type of cancer? What's the normal function of B cells?
The normal function isn't it to
Yeah, what's the what's the normal function of B cells? Guys,
Isn't it to carry like red blood cells?
no, B cells like what we talked about this morning.
Oh, those guys.
I'm sorry, B cells. What do you think? Zena, what do you think? Now that, let me ask you again, what do you think they are now?
Oh gosh, I just had it.
They produce antibodies, right, to fight bacteria.
Right.
That's what I was thinking, but it won't come out.
Okay, so they're the ones that are going to normally produce antibodies against antigens. They're going to proliferate. They're going to get activated by an antigen that they bind to. And then they're going to produce some plasma cells, mostly plasma cells, and then a few memory cells. But in this case, they're not doing that at all. They're just proliferating. Whether they've gotten an antigen that's activated them or not, they're just proliferating. Despite his apprehension and low spirits, Jesse showed up to the lab and was hooked up to a machine similar to the one used for his chemotherapy. This time, however, Instead of the machine administering drugs to him, the machine removed his blood and seemed to send his blood right back into his body. Dr. Sanguine explained that the machine was removing his T cells, so he didn't have to get them from the bone marrow. They didn't have to get them from his bone marrow. See, I didn't know this. I honestly read through the questions and I didn't read through what it said in between. Removing some of his T cells, and they would be reprogrammed to help recognize and destroy his cancer. Jesse wanted to know how T cells worked and how they would be reprogrammed. We're not so worried. I'm not going to ask you to be able to understand reprogramming and genetic engineering. But let us review how a normal CD8 cell first recognizes an antigen presenting cell and undergoes T cell activation. Below, draw two cells, a CD8 plus T cell on the left and a professional antigen presenting cell that recognizes it on the right. Include the proteins present on the surface of the naive CD8 T cell and the proteins on the cell it recognizes. Remember to include important co-stimulatory and signaling molecules involved in activating naive T cells. Label the proteins on the surface of both cells and include the receptor-associated proteins that become phosphorylated. We're not going to include the because we haven't talked about phosphorylation and T cell activation. We're not going to worry about this last part. Okay. Label the proteins on the surface. We're not going to worry about including these receptor-associated proteins. That doesn't matter for our studies right now. If we were taking a semester of an immunology course, we'd learn all about that, but right now we're not going to. So looking at this. Now, is it here? So here's your, let's see, B cell activation. No, it's T cell activation. Sorry, I'm getting a little tired and I apologize. T cell activation. Here's your T cell. It binds to an MHC molecule. Is it an MHC1 or MHC2 molecule? Look at the previous slide, and it tells you that it binds to an MHC1. So what I would do is I would give it a picture like this. I wouldn't draw any more than that because we don't have that we don't know that much about it, right? But we do have access to this. And then you could draw how and explain the process of how it's activated. Put this in your own words. Questions?
How many points are these words? Because I feel like I'm gonna suck at them.
It's worth the 10 points.
For all of them? For the 3
Yep.
of them? Okay. All right. Thank you.
And I got a comment last night, and because I've noticed it's a major issue and it's not your fault. I think it's partly because this is an online course. I think for lab, I need to start because Wiley is never going to give you deadlines on D2L. And the only place where they're posted every time is on the syllabus. And I have worked to keep it constant. Nevertheless, people are still confused as to when things are due. And so I will try to start putting something out every week in Lab about when what's due the next week.
I have a question. Do you want all 17 of these questions?
No,
answered?
because we're not going to answer all of them. We're not going to just answer the ones that we're answering.
Yeah, I'm following along. But okay, so you'll let us know what is due at the end.
Well, yeah, no, if we've answered them here, so you want to answer all of the questions for the IDA, that iron deficiency anemia.
Yep, all
You
of
wanna-
them.
Yeah, yeah,
Okay.
we went through all the answers.
Yeah,
here.
Yeah, I know.
Yeah,
I'm like setting up a Word document.
I understand. I understand.
Just want to.
Let me come back.
Thank you.
Nope,
Hey, I have a question
You're-
too. I'm really sorry.
go.
So, back to the like the scheduling and the syllabus thing. I was looking at my grades and I realized, I never did the Wiley blood lab, but I think it's closed. So if you're able to open
You
that, I would
will-
really appreciate it.
When do you think approximately you'll have it done, Lauren?
I can do it after class.
Okay, I'll set the deadline for you for Sunday, all right?
Thank you.
Oh, I'm
Sorry.
sorry. I also sent a message as regard to that.
Was it today that you sent it to me,
No,
Oyo?
no, last week.
Oh, I apologize. What was it for?
Blood smart, smart book, something.
Was it the Wiley blood?
Correct.
Do you think you could have it done by Sunday?
Sure, I can do it today.
Well, as soon as class is over, I'll extend it both out for you and Lauren. Okay,
Right. Thank you very much.
you're welcome.
Professor, please, I have a question too. When do you want us to submit the case studies?
And they should be submitted by next week. So next Wednesday at midnight or next Thursday at midnight.
Can you please show me how to get to the to it,
How to what?
to locate the case study?
They're under the case studies. I'll show you in 1 second.
So they're actually not under like the case study. You have to go to the
No,
Wiley exercise and you have
that probably
Sure.
would have been, I kept thinking where to put them, and I put them under lab because we're doing them in lab.
yeah, it's under blood and immune system case studies, but you have to like click under the Wiley lab exercise and scroll down a few
Do you see it, Toyin?
blocks.
Toyin, do you see it?
No, I'm still trying to get it.
Well, can you see it on my screen?
I had the same issue yesterday. I wasn't able to open it because I couldn't even find it. I went under the case studies too, but I wasn't able to
Why?
like locate them. It won't open up for me. So
Did I
guess-
send you guys an email saying where they were located? And they weren't, were
yeah,
they not?
you just
You
say
were-
that to
It's
review them.
confusing. It's not you it's just confusing. It's not you that made it confusing. It's just confusing. So once you find it, then it's easy.
Okay,
Does that make sense?
kind of. Toyin, did you find it?
Yes, yes.
Okay,
So both of them we have to do by next week?
well, yeah, and we're going through the questions. I'm giving you the answers right
Yeah.
now. And so I'm not expecting you to do a lot of extra work outside of class. Do you understand that?
Yeah.
Yeah, let's continue, but realize what we don't get done today, don't turn in. I don't want you to, some of this you're not going to be able to find. You realize how we're searching for the answers. Some of them you're not going to be able to find unless you really unless you knew a lot about this, which you don't. But some of this you do. So yeah, it's all due, but I'm giving you the answers right now.
Okay, one more question from me about that. So the extra credits are like unlimited time, but the case study is due next week. Am I
Right.
correct?
The case studies is not extra credit.
Okay, okay,
This is essentially a lab assignment
gotcha. Okay. that makes
because
sense.
as you noticed, we did the heart last week in lab,
Yeah,
and you recognize that we haven't even touched on the heart in lecture yet. And I felt like we kept getting ahead in lab, and we're not anywhere near that in lecture.
right.
And I didn't want to, I wanted to take a day, first talk about case studies, what you'd see in the future. And sometimes this is a good discussion to have in class. Does that make sense?
Yeah, it does.
Rather than just keep going forward with the material and people not knowing what to do with it.
Gotcha.
It's okay for labs to sometimes be out of parallel with lecture, but not, we're just getting more and more ahead in lab. And so I thought, take out a lab and put in case studies and get a little caught up in lecture.
That's a good idea.
Okay, so let's see here. In your drawing, does the T cell recognize its target in an MHC dependent manner or independent manner? Why would you think it would be MHC independent? What would it mean to be MHC independent? What would it mean to be MHC independent? If you're independent of something, what does that mean?
Would it mean that the T cell it doesn't need the MHC to recognize the
Yeah, it doesn't need it.
the infected
Right,
cells?
right, Martin, it doesn't need it. Does it need it in this case?
Yes.
Yeah, so it's MHC dependent. Because how are T cells activated? They have to bind to an MHC molecule. They're not like B lymphocytes that don't require an MHC. This is cell-mediated immunity, right? So you have to have direct contact with the cell, and that occurs through the MHC molecule. Does that make sense, Zena?
Sorry, yes.
Okay. So the CAR that will be expressed on the surface of just these T cells is a genetically engineered protein. It contains various domains of different proteins with 3 goals: to bind specifically to the intact protein on the surface of BALL cancer cells, insert in the plasma membrane of T cells, and help activate naive T cells. Let us investigate each of these domains and their roles. This is where we're going to skip through some of this stuff. Bear with me here. BALL cancer cells recognized are identified by a present on. Okay, what protein are they recognizing on, excuse me, what protein are they recognizing on his cancer cells? What marker? This picture up here, what marker are they recognizing?
Is it the peripherin?
Nope, nope, that's an enzyme that's gonna, remember that's the enzyme that's gonna poke holes in the cell that it's gonna kill. What do you think this CD19 is? Do you think it's a protein? Okay, so looking at this, an illustration of an engineered T cell recognizing and attacking a leukemia cell. The CAR molecule binds to the CD19 on the leukemia cell. This activates the T cell. That's it. Remember what CD19s are. They're glycoproteins. It's a protein. Do you see that? Okay, someone tell me yes or no.
Put a point to it.
So we can see it.
Say it again, Martin.
So would you be able to point to it on this, like on the picture?
Well, I can't, honestly, I can't tell you looking at this picture specifically what it is. I just know that's what it is right there. That's the answer.
Okay, so the CD19 is the
Yeah.
glycoprotein.
Right.
Okay.
And now the next question that this thing asks is this protein present only on cancer cells? What do you think? And before we answer, I'm gonna look. I wanna Google CD19 and see what it actually says about it. CD19 and leukemia. Okay, so CD19 antigen in leukemia and lymphoma diagnosis. The CD19 antigen plays an important role in clinical oncology. In normal cells, it is the most ubiquitously expressed protein in the B lymphocyte lineage. So one way that you could find the answer to that question is to Google it like I just did, right? So now we know it's expressed in the B lymphocyte. So it's normally expressed everywhere. Ubiquitously meaning everywhere. CD19 is induced at the point of B lineage commitment during the differentiation of a hematopoietic stem cell, and its expression continues through pre-B and mature B cell differentiation until it is finally downregulated during terminal differentiation. So it's downregulated in later plasma cells. I won't ask you in the ones that I don't discuss with you, the case studies that I don't spend time discussing them with you, or as much time discussing them with you. I'm not going to ask you to tell me about CD19. It's only in this one that we're using it for lab that I'm expecting you to know it a little bit. Because we know now it's a marker for, let me simplify what he's saying here. CD19 is a marker of differentiating or naive B cells. It will be downregulated in when that B cell becomes activated, and some of them become plasma cells and some of them become memory cells. However, because we're a B cell tumor, expression is maintained in the B cell lineage, because guess what? Most B cells, most tumors are undifferentiated premature cells. So wait, let me look a little bit further here. All right. So what is this protein? It's CD19. Is this protein present only on cancer cells? It is present on it's a hard question to answer. Because it's found on premature B cells that are naive B cells, they haven't been exposed to antigen yet. It would be present only on naive B cells. And since most cancers are naive, undifferentiated cells, you're most likely going to find it on cancer cells. What is the normal role for this protein? It's a marker for naive B cells. It's a cell marker, cell antigen marker for naive B cells. Does that make sense?
It's a lot, but yes.
Let me say it one more time too. So the protein is CD19. This protein is only present on cancer cells. Is it only present on cancer cells? Because CD19 is only found on naive, unactivated B cells. And most cancers are undifferentiated cells. So CD19 is only going to be found on cancer cells because it gets downregulated when that cell's activated to become plasma cells and memory cells. Does that help? Somebody tell me yes or no when you're ready. Zena, does that work?
Can you repeat that one more time?
Yeah. So the protein is CD19. And it's not only present on cancer cells. It is found on naive B cells. When we talk about naive B cells, we talk about B cells that have never been exposed to the antigen that's going to activate them. And because cancer cells, when B cells get activated, what happens? They differentiate into plasma cells and memory cells, right? Remember that from that clonal selection? They proliferate and then some of them become plasma cells, some of them become memory cells. In the case of this protein, it's only found on naive B cells. Well, most cancers are only undifferentiated naive cells. So no, it's not only present on cancer cells, because it's a marker of naive B cells. That's another way of saying it. So it's not, one more time, because I'm saying it multiple ways. This protein is present on cells besides cancer cells, because it's a marker that's found on all undifferentiated naive B cells. Once the cells get activated and they start to proliferate and become either plasma cells or memory cells, this protein is downregulated. So its normal role for this protein is to be a marker, like a cell marker or cell identification tag, to say this cell is a naive B cell that has never been exposed to its antigen that it was made for yet. Does that work?
Yeah.
Okay. What is the origin of the extracellular domain used to treat BAL? It is an antigen binding site that's going to bind CD19. So if you look at this question, it says, draw the complete molecule that is the original source of the extracellular domain of this antigen receptor, this chimeric antigen receptor. Label your drawing and circle the part of the drawing used to build the extracellular portion of CAAR. The easiest way to do this is looking back here. This is it. But this part right here, the antigen binding site, is going to be the CAAR. And this is, instead of just binding any antigen, its antigen that it binds is CD19. So this is the CD19 binding site for if this was a CAAR molecule. And now, I mean, I understand, and I'm slowly learning. If you wanted to, you could copy and paste a picture into your Word document. I'm not gonna, I know that sometimes people don't have the software to draw things out and scan them in. So you could take a picture of this or take a picture from the internet of a TCR receptor, T cell receptor, and say that this is the part that would bind to the CD19. Because that's the antigen for this CAR receptor that we're talking about.
Do you want us to cite the source if we do that?
That would probably be a good idea.
Yep,
The
that's
thing
fine.
is, if you cite the sources, then I don't question you where you got it, because that's where you got it.
Understood.
Okay. Transmembrane domain that allow it to be inserted. The CAR molecule contains a transmembrane domain that allows it to be inserted into the plasma membrane. The cytoplasmic portion contains an intracellular domains that can trigger the activation of naive T cells. What proteins are the origins of these domains? Bear with me just for a second here. You guys skip this question. We're not going to worry about the number 8. Okay. If we were in an immunology class, that would be great, but we're not. And if we were maybe in a graduate level immunology class, that would be great, but we're not. So we're not going to worry about that question.
I haven't gotten all the answers like to the questions that we've done so far. I mean, I got most of them, but like I don't have some of them. So like, and I know this is due in like a week. So will we be able to like get these answers on our own?
I think so. And you know, in all honesty, do the best you can. And I'm going to grade this thing on completeness. I'm not going to grade it on specifics. When I give you other case studies, this is a harder case study than what I'm going to give you. Some of it is, some of it isn't. But this, I'm going to grade this one on what you submit. And I'm not going to grade you for whatever answers you have.
Yeah, because
I don't know if I should have given that away, but I did anyway. I'm not going to grade this harshly.
I just created a template for the questions. If anybody wants it, I can send you a Google link if that's acceptable, Professor Gessel.
Mm-hmm. You know what, Claire? I don't care how you do it.
No, I'm just saying, like, it would save everybody, like,
It would. It would, and that's fine. That's perfect.
Claire, if you don't mind, I'll send you mine. I'll be the first one.
I have to switch devices when I do it, but I'll do it before the end of class.
One thing is I like in online classes, it's hard for you to work with your peers, but if you can, you should. For anything, practices, the case study that's coming up. The only thing when I ask you to do your own case studies, you can still work with others. It's not an exam. And I don't even stop you from working with others with an exam. But I want you to use your own words for explaining things. Below, draw 2 cells, a BALL cancer cell on the right and a CAR T cell on the left. Include and label the proteins on the surface of the cancer cell that are recognized by the CAR molecule. You guys, what 2 proteins are they? What's the major one? The CD19, because it's what's found on the cancer cell, right? CD19 that's going to be found within an MHC1 molecule. And that's what's going to be recognized by the CAR molecule, this antigen receptor. On the CAR T cell, draw a CAR molecule, label the domains, and indicate the antigen binding domain. So this is almost the same exact question as this one right here. But it's asking you to put them together.
Are there examples of what cells are supposed to look like when we draw them?
The PowerPoint,
Okay.
this is one right here.
All right.
Okay.
So I have a question. I had to step away for a couple minutes because I have a
Oh,
puppy.
Becky. Okay, go ahead.
I had to step away for a couple minutes. I have a puppy that was going crazy. So
Okay.
this will all be posted. This whole lecture will be posted so I can go back and rewatch
Yes.
and get any
Mhm.
information that I missed.
Yep, and
Okay.
Claire offered to send people a link of the word formatting.
Made a
program.
template questions. It's all set up. Have to answer the questions in your own words, but I'm sending like a blank, you know, the question will be in there.
Okay, much appreciated. Yeah,
Don't
He
have to
was-
set up.
yeah, sorry about that. I had to step away.
Well, and that's something else that I will give you when I give you a homework case study. That's not this one. I'm gonna copy and paste it into a Word document.
Much
And
appreciated.
therefore I can take out questions that I don't think that I want you to feel like you have to answer.
Also, Becky, minus question 8, I'll put that in the template. You don't
Yeah.
have to answer.
We're not doing this one.
Is that the only one that we don't have to ask? We
Yes.
don't have to do. Tariq was, I guess,
Right.
asking.
So far, yes. The other one is, there's this one. If you want to, Claire, take this out of your template. Include the receptor associated proteins that become phosphorylated. We're not going to worry about that.
Minus question 4 or part of question 4?
Just that bottom part of 4, this part right here.
Okay.
Because you guys don't know that stuff. I could probably look it up in a book I have, but I don't know that stuff right off the top of my head. So this is number 10, when car molecules interact with their extracellular target, this leads to a biochemical change to the intracellular domains of the car protein. What are these domains called? We're skipping this one too, number 10. We're not doing this one. Okay, so don't do number 10, don't do number 8. Does the CAR T cell recognize its target? Didn't we already explain this? Maybe I'm not such a bit, yeah, right here. But this was a normal, you don't have to answer that one either. This is for a normal CD8 molecule. Number 5 here. This one down here is asking about the CAR. I guess you could. Is it number 11? Does it recognize its target cell in an MHC dependent manner or an MHC independent manner? This is probably the easier question because we asked it before. What does it mean to recognize its target cell in an independent manner?
That means like it doesn't have to depend on that protein.
Yeah, it doesn't have to have an MHC molecule attached to it. So does this CAR T cell need an MHC molecule to be present? Now, go ahead, Martin, go ahead.
Yes.
I would say yes. One thing that I'm going to look really quick is I'm going to say, is CAR T MHC? Because it's possible that CD9, I don't think so, but I'm just curious what it's going to tell me. Independent. Is it independently of major? Okay, now we're getting into harder stuff because it says that it does it independently. We're not going to worry about it. We're not doing question number 11 because I don't want you to write down the wrong answer. And we're not going to worry about it. Because we're not masters of CAR T therapy. We're just using it to study immunology. The questions that I give you are not going to be if we haven't talked about it in class, I'm not going to ask you to do it. And we're not masters about CAR T therapy. So don't worry about question 10 or 11. Will the CAR only bind to cancer cells, or will it bind to normal cells as well? What would you say? What does it bind to, guys?
CD19?
Yep. And where is CD19 expressed only on cancer cells?
No.
No, it's not. Where else is it expressed?
Premature B cells.
Or let's call them naive B. Tiara, you're right. Let's call them naive B cells. B cells that have not been selected to proliferate into plasma cells and memory cells. And with that one, you don't want to answer. Go ahead.
Sorry, that was by accident. I was talking to my dog.
Okay, you don't want to answer just yes or no with that one, but explain where, first off, we know where it's expressed. So it's not going to only bind to cancer cells. It's going to go after naive B cells too. How is the gene? Okay, we're going to avoid this. Question number 13. So far, Claire, since you're the one who's writing all this down about which ones are not there, which ones you don't need to worry about, 8, 10, 11, and 13, we're not worrying about.
Got it.
Okay.
And that bottom of 4, right?
What? 4?
In the bottom of 4,
Yeah.
the last.
Yeah, not that bottom part of 4, right. Okay. After a few weeks, Dr. Sanguine informed Jesse that his therapy was ready and it was time to start with treatment. With the support of his family, Jesse had been trying to be feel more optimistic. However, Dr. Sanguine could tell he was still very apprehensive. So she tried to be positive and encouraging, wanting to make sure that Jesse knew what to expect with the treatment. She shared that they would be watching him closely for a few weeks, as there could be some significant side effects. Once all his questions were answered, he was ready for his infusion of CAR T cells. At first, nothing happened. Around 10 days, let me interrupt while I'm thinking about it. Claire, if you will send me, if you don't mind, if you'll send me your Word format document, I'll email it out to everybody.
Okay. You want me to
Can you attach it to an email and send it to me?
except I'm in Google Docs.
Oh, never mind. Never mind. I can't do that. Never mind. Sorry.
Yeah. Yeah.
So at first, nothing happened. Around 10 days, Jesse began to feel very ill, almost as if he had the flu. He had high fever, chills, and his blood pressure dropped to dangerously low levels. He feared the cancer was winning, but Dr. Sanguine once again assured him that these are signs and symptoms resulting from the treatment, not the disease. And his doctor was right. A few weeks later, the fever and chills were gone. So was the cancer. There are 3 different types of different types of T cells. including CD4, remember those are helper cells, and CD8 cells. What types of T cells are CAR T cells? What do you think they are? Are they CD helpers? Are they, excuse me, helper T cells? Or are they cytotoxic T cells? Look back at this. Do helper T cells actually kill cells?
No.
No, you're right. So like Martin said, they actually don't kill cells. They're kind of the little coordinators and tell everybody else what to kill. So they're cytotoxic T cells, they're CD8. And what will occur when these CAR T cells encounter their targets? Well, they're going to bind to the CD19 molecule, and then they're going to secrete some perforins and destroy the cell.
Can you say that again? I'm sorry.
Yeah.
So they bind to what?
I'm gonna do something else. I'm gonna look at this. So rather than it being a virus infected cell, it's gonna, the CAR T cell, say, instead of saying cytotoxic T cell here, say the CAR T cell is gonna recognize and bind to the specific CD19 marker antigen. When that happens, it's going to then release chemicals that destroy the cell. Does that make sense, Zena?
Yes, yes, thank you.
Let me say it one more time. I know that there's a lot of reasons I do a lot of repeating in here. And part of that is because I know that some people, internet issues are really funny. And so let me just repeat that. So the cytotoxic T cell is going to recognize and bind to the CD19 molecule on the cancer cell. Afterwards, the next thing it's going to do is that CAR T cell is going to release chemicals that are going to destroy the cancer cell. It's going to release like perforins and destroy it. Or you could even use this instead. It's the CAR T cell is going to bind to the target, and then the CAR T cell is going to release a bunch of perforin granules that are going to fuse with the plasma membrane, and the granules are the perforin is then going to insert itself in the plasma membrane of the cancer cells and lyse the cell, lyse the cancer cells. Questions? Do you feel like your heads are going to explode?
Yeah.
All right. I think we have about 30 minutes left. And I don't believe it's going to take us very long to work through the last 10 slides of the immune system. There's some discussion questions. Actually, let's look at the discussion questions on the PowerPoint. Bear with me. really quickly here. I'm gonna shift gears. So do you know what questions you need to do of those case studies? Do you want me to repeat them? Do I need to repeat them?
It's all of them, but 8, 10, 11, 13.
8, 10, 11, 13, and 15. Don't do 15.
Okay.
8,
And the
8,
last bit of 4.
yeah. 8, 10, 11. And 15. Don't do 15.
Did you say
You said
15,
13 too. 13 and 15 too.
13 and 15?
Don't do
Yes.
15.
Okay.
Let's do one last thing. And I can't remember exactly what the questions are. But it'll be interesting all the same. Skip through some of this. Question for you. Antibacterial soap, and you don't have, we're going to discuss these. I'm not asking you to answer them and submit answers. Or should I just let you guys go as you're all writing down answers to the questions? Should I let you go?
Whatever works.
Why don't I let you go? Do me a favor, look at these questions before we have class next Friday. They start on 105. Look at these questions and think about what you think this could be and what you think could be the problem. And how you think you would answer these questions. And I will see you next week.
All right. Thank you. Everybody have a safe weekend.
Thank you.
You too.
Professor, are you going to post the video?
Oh, sorry.
Professor,
Oh, you know what? I will try to post it. It has to process it first, and it'll take about an hour, and I'll
Okay.
post it after that.
all right, thank you.
You're
Also,
welcome.
Professor, just to remind you, it's on Blood Smart Book, chapter 18.
Okay, the smart book,
Right.
chapter 18. It wasn't Wiley. It was the smart book, or was it
Correct.
both? Only
smart-
was it both?
No, just not book, just the Smart Book.
Okay, all right.
Thank you. Thank you.
I got it.
Right. Thank you. Bye.
Bye-bye.
Thank you, Claire. I just got your link.
No, I think you guys all sent me your addresses. If you didn't, just because there's only a few of us left here, if you did not send me your address, what I'm gonna do is I'm gonna mail you some urinalysis strips. And as soon as I got the letter in the mail, I shared your addresses. I don't hold on to them. And I need to go through and write down everyone that I have, which I think I have the majority of you. All right, do you guys have questions for me? Would
No,
you
I
like
just
me-
want to make sure, like, the due dates, did you say you was gonna put them somewhere to make sure like we were all?
I will, I have to write it in, and what I'll probably do, it'll take me a little while to do it.
I'm sure.
But if, right, bear with me here. Right at almost the beginning, and I think I'll have to look and see just how this is gonna work out. I probably won't do it for the stuff that we do in the chapters, because all of that stuff is posted on your content page. But for the labs, right here, there's a place where I can actually write a description and then add describe. I'm gonna
Okay.
put it in there.
Okay. Underlining.
I mean, once I get started, it won't take me very long. I just have to do it. I think it's gonna help a lot of people. And other more experienced teachers do it all the time, but they're more prepared for their classes than I've been prepared for this one, because they've done it for years and I haven't.
Yeah. Well, you're doing a great job though.
So
You
Thank
are, you really
you.
are. I mean, you're